Now let's move to the next session, that is panel discussion. So I will request all our panel members to come on the stage and please take your seat. So in the panel discussion, we have Dr. Venkata Ramana, Chief Scientific Officer of Reliance, and Dr. Sunit Maity, Senior General Manager, Syngene, and Dr. Yaron, and Ms. Stacy. So I welcome you all to please come on the stage and take your seat for the panel discussion. Please, please. So we have already introduced Stacy and Yaron. Let me introduce Dr. Venkata Ramana.
You can cut down the introductions.
Yeah, maybe, yeah, yes. So Venkata Ramana is Chief Scientific Officer of Reliance Life Science. Dr. Venkata Ramana has obtained PhD in Biochemistry from CFTRI Mysore, and for the last 22 years he is associated with Reliance, and his expertise lies into molecular biology and cell line development. Next panel member is Dr. Sunit Maity, Senior General Manager, Syngene International. Dr. Sunit was trained in developmental cardiovascular biologics in Leuven, Belgium, where he received his PhD. He was involved in target identification using model system as well as CNS development. He has contributed towards the development of biologics, including recombinant proteins. So we'll start the panel discussion. We have a couple of questions for the panel members. So first we'll start with the cell line clone, then later we'll move to the process development. So I have a question from Dr. Venkat about the cell line. So just as we understand that cell line development is the key, and we face a lot of challenges in establishing the stable clone. For that, we require single cell clone because that is the regulatory requirement. So we have a question about the single cell cloning. What are the advances in single cell clone development, and how can we develop a high producer and stable clone using different tools and STP devices? Dr. Ramana.
Yeah, that's an important question when you try to develop a good cell line. So we need to show a proof that the clone that you finally selected is based on single cell cloning. Traditionally, people used 2 rounds of serial dilutions, limiting dilutions, and very rarely we have proof of clonality there. But recently, technology has improved. So now we have single cell dispensers are available. Of course, you can do without that also. You still can do limiting dilutions. But I think most importantly to me is imaging, whether you'll be able to image a single cell from day 1 onwards. Typically, I like doing single cell imaging every day until day 8, so that you can show cell is dividing between 1, 2, and 4, and so on. So that imaging really helps, and you will have very less questions from regulators. Thank you. I'll expand later if there's a need. Sunit.
Now I have a next question. Thank you, Dr. Ramana. I have next question to Stacy about the media and feed development. What are the current approaches for the right media development? You have already presented the case studies, but if you talk about the omics AI approaches, what is your opinion about that? Can we expedite the process development, media feed development, and can develop a good quality of the product?
Yeah, I think that, can everyone hear me?
Yeah.
Okay. I think that we're making a lot of advances, especially in using omics tools to really understand the cells and what they need when they need it. So I think, really starting with your clone and having that in place, and then we have a lot of understanding of media, especially with CHO cells, what is important for the CHO line, for both the growth medium and the feed. And then taking it to the next level, starting to incorporate these omics, where we're combining this knowledge we have of the media and feed, and also this more in-depth analysis of the cell line to really start optimizing what the final formulation will be. And as these tools continue to advance and we continue to become, they begin to be used more frequently, I think that's going to just continue to expedite how quickly we can get formulations developed. So I don't.
Yeah, thanks. Yeah, Yaron, can you please share your experience in media development, especially when you screen the media at a small scale, then when you go for the small scale tool, like say shake flask, then bioreactor, what are the challenges which we may face while process development and media and feed optimization?
Yeah, no, so related to use of multiomics, like you mentioned, I think the main advantage is that it doesn't deal with a specific clone or a specific process. It incorporates all the research papers that are done before to build this model, and all the metabolic pathways that are modeled are not for specific, they try to find generalist factors, general components that are influential for CHO cells. And then when you use this model for a specific clone, then you get highlights of what's really important, and it covers basically a lot of general information. And to your question about scaling up, that's another issue, because when you scale up, many things change. So I think omics for developing media, that's something that it's not going to change when you scale up. The media component usually will be fine, but later for process parameters, optimization, then you need to apply omics on different scales. But for media development, it will work fine on even small scale.
Thank you.
Maybe I can add on the scale-up part here. When you are developing the scale-up from 10 liter to larger scale, maybe we need to look into the KLA of the parameter, like what is the KLA, and KLA needs to be kept constant when you are sticking to the same bioreactor design. But if you're changing the different bioreactor design from 10 liter to 200 liter to 500 liter, then KLA would be our guiding principle, because the oxygen transfer rate or oxygen utilization rate plays a major role when you are doing the scale-up. So KLA is a major parameter beside this metabolomics approach, what both Stacy and Yaron talk about. We need to look into the KLA too.
So, Dr. Sunit, what is your opinion about other parameters like PYV or maybe TP speed? Because these are the scale-up parameters which we consider when we scale up the process from small to large scale.
Thanks. That's a good question. Yeah. So beside the KLA, the PV speed and all those, but another thing what we also should focus on the DOE-based study, as all of us are talking about. When you are doing the scale-up, you try to figure it out what is your process parameter, what you want to standardize. So if you do by PV or impeller speed, different agitation, KLA, what we all spoke about, that will define what kind of scale-up parameter you need to look into it. And as both Stacy and Yaron also spoke about, that we need to do a multivariate analysis. Because if you do the multivariate analysis when you are developing the process, that will reduce the failure. So the failure will be much less, and scale-up parameter would be much more better when you have carried out this statistical-based tool. Another thing what we all need to remember when you are going to scale up to the commercial scale, process characterization plays a major role. You need to characterize your process parameter, because that will give you the confidence of the robustness of the process and also different parameter. And generally, what we do the process characterization by doing a scale-down model. So everybody who is trying to develop a process in the commercial scale or the clinical scale, scale-down model is absolutely important. Process characterization, KLA, P5V, tip speed, these are all plays a major role whenever we are doing the scale-up of a process from 10 liter scale to the commercial scale.
Good, thanks. Can you please throw some light on the PACT? Because that is also very important when you talk about the process. You nicely explained about the scale-down model and process characterization.
So process analytical technology, that is a major thing what we all need to integrate. Maybe you can do the Raman spectroscopy, online monitoring, real-time monitoring the process metabolites. Whenever you are developing a process from scratch, include the QBD approach, PAT approach from start if it is possible. There are various tools of Raman spectroscopy, look into the metabolite, or you do HPLC analysis you can do. There are some software called Rebel, which are all available right now. Do it from the beginning so that you can look into the different parameter, how it is changing when you are developing the process.
Thanks. I have next question from Dr. Ramana about the protein expression. What is the next in cell line development? People talk about 2 gram per liter, 5 gram per liter, 7 gram per liter. What is the next? And what are the challenges? One side you have, upstream process, other side you have downstream process. Cell line developer get a lot of inquiries, lot of, questions about the quality as well as, stability and the protein expressions. What is the next?
I think we have evolved now from DHFR-based expression systems to GS. Now people are talking about transposon-based expression systems. And I believe probably that's the latest. And what I've seen is that probably gives you one of the best results in terms of yields. I think we should also remember, I think a lot of people have stressed here that cell line can only produce, to a certain amount of protein. The rest of it is actually depends on how you optimize the medium. In our hands, I think we have doubled the yield from cell line to final fermentation. So if somebody claims that this transposon system can give you, say, 7 grams, 8 grams, so you have the potential to make it to 16 grams. And it's very interesting that the cell line development is never a static process. It keeps developing, and a lot of industries, of course, industry players keep investing in these company, in these technologies to come up with new technologies. So to me, transposon technology is very interesting to see what it gives us. So let's hope we'll get multiples of tens. Thank you.
Maybe I can add what Venkata sir was telling. There is a paper published by BI using transposon-based technology from transfection. They have initiated clinical trial phase 2 in 3 months. So that's an amazing achievement. We cannot even think of developing a cell line in 6 months. They have initiated phase 2 clinical trial in 3 months' time from day of transfection. That is the transposon-based technology.
Yeah, that is possible when you have high throughput devices and,
they're doing from the stable pool. Now the regulatory government bodies are allowing to initiate the phase 1, phase 2 clinical trial from the stable pool. So that's why they could do it in 3 months' time.
Next question I have from Yaron and later Stacy about the media, switching the media feed. Suppose we have developed the process and we reach to certain stage, clinical trial, phase 1 or phase 3. Now we get good media which gives better protein expression and better quality. What is your opinion on that? Can we change the media in between?
Yeah, no, it's a great question. So we had customers that changed media already in the clinical trials phases, and it's okay to do it. It's a kind of a process change, but there's a lot of reason why customers want to do it. And one of the main reasons is that the original media that the clone was developed in is usually old. It's a long process. It could be like 10 years old formulation of basal and feed media, and new development in new feeds that emerging now makes a really increased titer, and also the performance is much better for new feeds. So that's the main reason for customers to switch to a new feed, even if they are late stage. Also, you have issues of supply, so media supply and the availability, especially in the pandemic, for example, or other issues in the world that can affect supply of media. So some customers prefer to switch the supplier to have more backup supplies also. And cost is also a big issue. So titer is important, but also other factors. Yeah.
Stacy, I have a question for the quality and titer improvement. What are the approaches, and when you have a final clone and developing the media feed supplement, how can we speed up the process, and what are the approaches to improve the titer and quality?
So I think it's really, first of all, understanding what your ultimate formulation needs to be. Do you need to be chemically defined? Or can you use hydrolysates? And once you understand that, then I think it's important to start with media screens. I want to add on to the older formulations. When I talked about sometimes there are negative drivers in your media, some of these older formulations, components get carried along just because they've always been there. And that's why sometimes really looking at your media, taking a fresh approach at screening some new media, some new feeds is a really good place to start, just to make certain that the formulation is better optimized to a bioproduction process. So once you get through some of these media screens, if you're using especially established media, commercially available media, then maybe it's looking at some supplementation to add on top of that. There are a lot of different options out there that you can add, but all of that has to be in working along with doing your process work in your scale down models to make certain that you understand it's not only that what media and feed you're adding, it's when you're adding it and how you're adding it, because shifting when and how you add these different components can really have an impact on all your product quality attributes. So I think that we now have a lot of different media available that are much better optimized for this industry, so I think that it really speeds up the optimization. But then I think it really is important to understand what are your requirements for your process, and then looking at the available components and starting your optimization there. And then I'll say it again, I think it's important to look at multiple lots of everything that you're looking at to make certain that you really understand the capability of your process and any variability you might see, so that you can be proactive in addressing it.
Okay, thank you. Now we can open session for a couple of questions, maybe 1 or 2 questions we can take up. So Sudhati and Dr. Saranya, can you please pass on the mic?
Hi, Nandakumar here. Okay. So this question is regarding the proof of monoclonality. So generally what happens is the monoclonality you do when you are screening your clones. But ultimately there is a time period of 6 to 7 years when you go for a submission. So with the blessings of AI and all those technologies, after 6 years you came with good technologies. And the current technologies where you do a single cell dilution or a printing also, they may look irrelevant at that point of time. So in that case, another outway is to have a proof of monoclonality or monoclonality assessment then. So how do you suggest it? Because that can be a bottleneck. After 7 years, the regulatory body says that the proof of monoclonality that you are showing me is not a good technology. So
that's an interesting question. Of course, technologies keep developing, but you always have to comply to the current regulation. So 10 years down the line, if you think the regulations have changed, I'm sure regulators will look at your cell banks in a very positive way. But I know a lot of companies actually change cell lines. They derive single cells again, and then do the process once again if required. But I'm sure regulators will not ask you to do because you have used old technology 10 years ago. But if you are deriving today, I think you should use latest technology. And if you have done that, and if you are convinced that this is a single cell, I think you should be okay.
thanks.
Maybe I can just add, if you have the monoclonality report, which all the companies are providing along with the clone select or Solentim and all those, that is the guideline FDA is asking for. So whether you do it now or 6 years down the line, that report should be sufficient.
With that respect, if you look at the cell lines that people have done by serial dilutions, today they may look irrelevant because of the availability of the newer systems.
Yeah, but that time the guideline was not there. Right now the FDA is absolutely compliant that you need to provide a monoclonality report showing what you're studying every day, taking pictures from the single cell till the 8 days what is suggested. If you document that, whatever the technology is using, it is proving that it is a monoclone. So that should suffice if you do after submission after 7 years also.
Okay, thanks.
So can you pass on the mic to Saranya?
Thank you everyone for your time today. This is Sharanya from Ipca Laboratories, and my question is to Dr. Ramana and Dr. Maity. So it's a continuation to the question that was asked right now, but there are certain cases when your cell line development has gone to a very late stage of development and everything else is pretty great. Is there any, is there a retrospective approach to prove monoclonality? Once everything, because recloning and repeating the entire process probably might not be worthy of the resources in terms of the company's point of view. So are there any retrospective approaches that can be used to prove monoclonality of an already existing clone? That is my first part of the question. Second part of the question is, are we taking this monoclonality more seriously than it should be considered, right? We know, like, once we show consistency of a clone, once we show, the robustness of the clone's performance, not only in the final at-scale process, but also, all the different during process characterization that is done at a late stage. So sometimes as a clone developer, I think monoclonality is taken a little bit more seriously than it should be from the regulatory point of view. But that's just my thought. I would like to hear your thoughts on that. Thank you.
So maybe I can,
first of all-
okay, you go. I'll take the second part of the question. So recently what I was telling you, there is a research article published where they have allowed, the FDA has allowed to initiate the clinical trial from the pool stage. It is not a monoclone. Exactly, based on the transposon-based
Right.
technology. And the phase 1, phase 2, you can do it from the pool stage. And phase 3 has to be done from the monoclone. So FDA is also thinking what exactly told, because if you can show the process consistency and the pool stage also, if you do lot of batches at the same time and it is showing the consistent specific productivity or the target product profile, then I think regulatory is also asking and understanding that it can be done from the pool stage if you show it is a consistent, robust process. So that is my feeling too. I completely agree with you on that.
Yeah, I think that's the answer even for the first question. But I would look at, how did you derive your cell line in the first place? Was it a pool? So did you dilute to a single cell and then developed, but you did not have the technology to prove it? So it's possible that you must have got a single cell, but very likely that the technologies that we used in the past, really they are coming out of a pool. Though maybe one cell must have survived, but many cells probably were there in that well. So there's no way to prove that. So one proof that you said is consistency. I think that's a great way to do that. But then there's no proof, again, from clonality. The other one, I'm just guessing, I'm hazarding a guess. What if you do whole genome sequencing? Would it tell whether it has come from single cell clone or not? I heard that it would tell, but I'm not certain. Maybe we can look at that aspect.
Yeah, this is good point.
Maybe
Nowadays-
I have another thought also. You can do a small single round of limiting dilution, because generally we get to see from pool to clone the titer increases. If you do one small round of limiting dilution and see there is a product titer consistency,
Right.
then we know it is coming
But
from a clone.
there's a lot of subclone variation that happens when we do
But if it is coming from the monoclone, then it should not have the variation.
No, but that's not how that is the ideal situation. But a lot of studies and personal experience has shown that subclonal performance subclones are completely different from the main clone, mother clone that it was derived from. But certainly whole genome sequencing and probably identifying the sites of integration and then looking for it is one possible option. I was just looking for your thoughts. Thank you.
We take one more question, maybe Siddharth. Okay, we have happy to see the excitement of our audience. I am really sorry, running time. Yeah, please. Siddharthi has raised the. Yes, please.
Hello. Hi team, this is Samir from Chemistry India company. So my question regarding clone stability. So as you are aware that clone stability generally take 4 to 5 months to ensure my clone is stable or not. So can we have any method that can predict within twice a week that my clone is stable or not? Because stability a lot of work in a cell line development. Thank you.
Unfortunately, I think there's no shortcut for checking the stability. You have to check over generations. And we should also do that in our own interest. After a few years, you realize the cell is not stable. You don't want to be in that situation. At least I'm not aware if there's any shortcut.
Right.
Okay, thank you.
Siddharth, you have question? Okay.
We can think of kind of markers which is available for the stability.
So there are kind of mitochondrial markers are available which can say that that clone is stable.
But at least still you need to do different passages, right? 60 generation you have to perform.
You have to.
No, I'm just adding on to that. Now my question was to Dr. Ramana, even though we prove it is cell is monoclonal, you know CHO cell's plasticity, you know that CHO cell when you passage is there are a lot of inversions and translocations happen within the single clone itself, you know. There are 21 or 22 chromosomes, and they if you same clone, single clone, if you passage is continuously for about 6 months, there is definitely the translocation and inversion event is going to happen. So how to address that? Then what is the relevance of doing the single cell cloning? Because there are a lot of literature available where it shows CHO cells is not very much stable in terms of the chromosomal translocations and inversion events.
Yeah, it's a good question, but then your interest is to look at the stability over generations, over limited generations. You're not talking about unlimited, right? That's why you make a cell bank, master and working cell banks, to maintain that generations. So you're not really worried beyond 100 what happens, right? You can study, but then it's of no relevance. Your interest is up to 30, 40, 50. If nothing happens up to that, then you're fine. You know, you're safe.
To add the same thing what Saranya has said. So like when you do the limiting dilution again for the single cell clone, you have a variability of 100% in terms of the productivity as well as the quality attributes. Then it's very difficult sometimes to have actually that relevance of that monoclonality.
Yeah, hard work.
Last question, very brief.
Yeah, can you pass on the mic, please?
Yeah. So to Sunit and Dr. Ramana, so what you see how you see the future of new expression systems, particularly vectors? What kind of future you see? As we know that we have all those established systems in terms of vectors, in terms of cell line. So, but there are some evolving systems also, like I came across one of the fungal expression system, Myceliopthora thermophylla. It's a C1 expression system. So, and then there is an Vibrio nitrogens. So, couple of those things. So, how industry sees those novelty in this domain, in terms of cell line and the expression system.
So yeah, good point. So the C1 expression system, they give a good titer about 10 grams per liter easily. But we need to check the quality of the molecule. If it is a biosimilar, it has to match. If the novel molecule is a great, it's a good system to look into it if you're thinking of a new molecule start with. You can modify the C1 expression system to have your own glycan. That modification is possible. People are also looking into the plant-based system, the Canadian-based company, I think Transpath or someone, who is developing biologics using the plant-based expression system. So yeah, it's being looked. But if you look into the biologics in the CHO cell line-based expression system, the transposon is really coming up very well because it reduces the screening of many clones. It is a targeted-based random integration. So it integrates at the multiple location single copy genome. So that is a new expression system which is coming up very soon. CRISPR-Cas9 has been also used for transfection but we all know the legal aspect of it. We do not know who owns the patent. So the commercialization of the CRISPR-Cas9 plays a major challenge for all of our companies who would like to use it for commercial purpose. For developmental purpose, R&D license, everybody can use CRISPR-Cas9. But for commercialization, that is a major challenge. Otherwise, CRISPR-Cas9, transposon-based system, C1 cell line, plant-based expression system have been also looked into it.
Yeah. Thank you very much. We conclude this session here. And thank you very much to the audience for your patience and listening us. And thank you very much to all our speakers for sharing information and experiences. Thank you. I'll hand over to Pooja now. Thanks. A big round of applause for all of you.
Hello. Thank you for the insightful discussion. I would now request Dr. Sanjeev Gupta to felicitate our speakers and panelists. Ms. Stacy Holdread. Dr. Yaron. Dr. Venkata Ramana. Dr. Sunit Maity. Dr. Ravi Mishra. I would like to thank Dr. Sanjeev Gupta and would request Dr. Ravi Mishra to felicitate him. Thank you all of you for attending the session. I would request all of you to visit our Biopharma exhibition at Grand Ballroom. The next session will start at 2:30 and the lunch will be served right outside this hall. I would request all of you to collect your coupons from Ranjit. He is just outside this hall. And please carry your bags with you since we have to rearrange chairs here. So sorry for the inconvenience, but please. Thank you so much. Have a great day.